Alright, welcome to part 3 of your notes. We're going to talk about probabilities and ratios, we're going to talk about incomplete and codominance. Okay, so learning target, the first one we're going to hit is applying mathematics to determine patterns of inheritance. I know, we're going to throw some math into science, but you know, it's life. Inheritance actually follows the rules of probability. Actually follows the rules of probability. So what are the rules of probability? Well, to demonstrate this, I'd like you to think about flipping a coin. How many of you guys have ever flipped a coin? Hopefully all of you. If not, grab a coin and flip it. You've got a 50-50 chance of getting a head, or getting the heads, or getting the tail side of the coin. There's only two sides, there's only two options. So you've got 1 out of 2 chance of getting the head, or 1 out of 2 chance of getting the tail side of the coin. Now, when we're dealing with genetics, so far we've only talked about genes that have 2 alleles, okay? And for the most part, that's when we're looking at genetics, that's what we're looking at. We'll talk later about there are some types of genes that have more than 2, okay? But that's for later. Anyway, so since we're looking at just 2 different alleles, you have a 50-50 chance of getting either the head or the tail side of the coin, or you've got a 50-50 chance of getting the dominant or the recessive version of the allele, which is why you get a 50-50, or why you have genetics following the rules of probability. Okay, so what we want to know is we want to apply this in offspring. So if I was to give you a Punnett square like this and told you that dad and mom are heterozygous meaning they have a capital and a lowercase letter, okay, and if I asked you what are the chances of one of their offspring being a homozygous recessive, okay, or homozygous dominant, you need to be able to look at this and go, okay, well, homozygous dominant, that means we're going to have 2 big B's. And if you take a look at the Punnett square, right here we see this is the only one that has 2 big B's. So I've got 1 out of 4 possibilities. So I've got a 1 out of 4 chance. Okay, what's my possibility of having a heterozygous offspring? That means heterozygous big B, little b. There's 1. There's another one. So heterozygous, we're going to have 2 out of 4 chance. Okay, or in other words, 50% chance, right? And then lastly, for homozygous recessive, little b, we're going to have 1 out of 4 chance. Okay, so that's what we're talking about when inheritance follows those rules of probability, because we have a 50-50 chance of having the big B or little b, but when you combine those 2 heterozygous individuals, these are the kinds of ratios that you're going to see. Okay, you'll need to know that for your learning target, your learning check. Okay, so you can convert these into decimals, into percentages if you want. You can reduce them. This one, you're going to have what? 25% chance, right? 25% chance. So if in the future you ever get your genes, if you ever get your karyotype and you do a genetic screening between you and your partner, then you can find this is where those chances of your child having some kind of disorder or some kind of trait, it all comes back to the Punnett squares and the work that Mendel did with his P's. Alright, so inheritance and probability, I'm not going to spend too much time on this because I think it's pretty self-explanatory. Okay, however, this is really important. You really need to make sure that you know that when you take 2 heterozygous individuals and you're looking at 2 traits, so a dihybrid cross, okay, you're going to see this phenotypic ratio of 9:3 to 3:1. That ratio is so important, and I can almost guarantee you that you're going to see it on future tests for my class, for an AP class, even AP tests. Okay, that 9:3 to 3:1 ratio is so important. Commit it to your memory. The other thing you need to understand is what those numbers mean. So just real quick, okay, that 9 means this is how many of the offspring you're going to have that are going to carry the dominant alleles for both traits. The first 3 means this is how many are going to carry the dominant allele for the first trait and the recessive for the second trait. The second 3 is how many are going to carry the recessive allele for the first trait and the dominant allele for the second trait. And then the last one, that 1, is how many are going to be recessive for both. Okay, so here's an example for you of how you get this 9:3 to 3:1 ratio. If you take a look at this Punnett square on the right side, okay, this shows you dihybrid cross, that you're looking at all the possible combinations that are possible for the offspring. Okay, so how many of the offspring are going to be yellow and round? Well, there's 1, there's 2, 3, 4, 5, 6, 7, 8, 9. Okay, so 9 out of those 16 are going to be round. Okay, oh look, we're starting to build our ratio. Okay, how many of them are going to be green and round? 1, 2, 3. How many of them are going to be yellow but wrinkled? 1, 2, 3. What do you think that last number is going to be? How many of them are green and wrinkled? Just 1. 9:3 to 3:1. You will see that anytime you cross 2 heterozygous individuals in a dihybrid cross. Okay, 9:3 to 3:1. This is a really good way to double check yourself when you're working on those kinds of problems. Okay? Alright, so here's your learning check. Oh, no, it's not. Your learning check is your next slide, sorry. Monohybrid ratios, that's like that first one that we talked about with the coins. Okay, for your phenotypic ratio, you're going to have 3 out of the 4 individuals showing the dominant and only 1 showing recessive. For dihybrid, you've got that 9:3 to 3:1. Now for your learning check, this one's going to be difficult. This one I want you to set up a Punnett square and do a trihybrid cross. Okay, so something like this. I'm not going to sit through, I don't have time to go through it, so go ahead, set that up, work on it the best that you can, and we will talk about it tomorrow in class. Alright, next learning target is number 3. We've been talking about Mendelian genetics. We followed his, the chromosomal theory of inheritance and his model of inheritance, talked about law of segregation, independent assortment. And of course, life's not as simple as that. Okay, we're going to talk about things like sex-linked, sex influence, multiple alleles, incomplete dominance, codominance. Okay, you're going to be able to solve some problems that are relating to those different kinds of models of inheritance that don't follow Mendel's models, and you need to be able to explain some of those deviations. So let's go through it. First one, this sign here means don't worry about writing this down. Feel free to, but don't worry about it. Okay, so Mendel's principles apply to all sexually reproducing organisms. However, okay, it's not that simple. So we've got a picture here of this black cat and this brown cat. So if we're following Mendel's principle of dominance, okay, one of these is going to be dominant and one of these is going to be recessive, which means their offspring should either be all brown, like this kitty, or all black. Okay, but what happened here? Okay, we get this calico cat that's got both brown and black. So let's talk about that. Okay, we've got incomplete and codominance. First one we're talking about is incomplete dominance. Okay, incomplete dominance, this is going to result in a blend of phenotypes. So an example of that would be if we had a red flower that's dominant that crosses with a white flower and that white color is recessive. Okay, so that red and that white combine, and if they've got incomplete dominance, you're going to see something new. You're going to see something pink. Okay, with the Mendelian genetics, you would have seen either a red flower or a white flower, because that is complete dominance. Okay, however, It's not what we see a lot. So we've got this incomplete dominance, which means you're going to see something new from mom and dad. New, that it's not going to be like mom, like dad. It's going to be new. It's going to be this pink color. Okay, then you got codominance. Codominance is going to result in both phenotypes being expressed. So that would be like the example with the cat that you just saw. Okay, if you've got a brown coat, and let's say that's dominant. Oh, I gave you an example with horses because that's one too. If you've ever seen a doggo, his horse is expressing codominance. Okay, brown coat, let's say is dominant in horses. I don't know if it really is or not. Okay, white coat, let's say it's recessive. If you have a brown male and a white female, or a white female, brown male, they have an offspring, you're going to get pinto because those genes are codominant, which means it's going to show both brown and white. Okay, another one that is more applicable to you and I is codominance. Oh, sorry, is blood type. I just realized I need to move the screen. Let me see. Okay, so we've got codominance here. We've got blood types. So A and B blood types are codominant, which means, if you take a look here, A means we're going to have all these little circle shapes here. Okay, for the antigens. B is going to have these square shapes here. Diamond, it's not really diamond, but the square-shaped antigens. Since they're codominant, if you've got AB blood type, it means you have both the square and the circle. O just got nothing. Okay, O is recessive. So, codominance. How do you figure out blood types? Okay, blood type A is dominant with blood type B, and that should say codominant, sorry. Blood type A is codominant with blood type B, and O is recessive to A and B. So, what are the possible genotypes? Well, if you've got a mom that has A, you can have AA, mom and dad. You're going to have AA. Myself, I am O-type blood. My husband's actually O-type blood. O is recessive, which means all of our children will always be O-type blood. Okay, so, now, hold on. Going back to that example, if I had B-type blood and my husband was O-type blood, that means our possible blood types would be B and O, or BO. Okay, if you have A-type blood and you get together and have an offspring with someone that has O-type blood, then they're going to have A-type blood. Okay, if you have any of these types of bloods with an O, if you have AO, that's just A-type blood. If you have BO, that's just B-type blood. If you have AB, then it's AB blood type. Okay, that becomes really important when you do blood transfusions because these antigens, your body has specific antibodies in the plasma that if they don't match with the antigens on the blood type, your body will attack those blood cells, which is why O is the universal donor because it doesn't have any of those antigens, which means whatever type of antibodies you have in your plasma, it doesn't matter if you have O-type blood because those antibodies are not going to attack O because O doesn't have any of those shapes on there. So it's going to kind of fly under the radar. Alright, so we got to get to the learning check. So here's your first learning check. Second learning check. Third learning check. Now obviously you're going to have to pause these and answer them yourselves, bring them to class, and we will discuss them there. Okay? Hasta luego.